Alright, hi, my name is Faith Forbes. This is my brother Austin and my dog Luna. She's in her schoolgirl outfit today because she's ready to learn. So for my project, I'm going to be talking about protein synthesis and how it's involved with RNA and DNA. And then I'm going to talk about genetic mutations. So to begin, DNA and RNA are both something that we call nucleic acids, and but they work very differently when it comes to the two stages in protein synthesis, and that's because they are different. So the differences between them is that DNA is what holds the genetic code, or it's kind of like the blueprints of the cell, so it's a little bit different from RNA in that sense, and it's double-stranded while RNA is only single-stranded, and they both have 4 nitrogenous bases each, but what their nitrogenous bases are a little different. So DNA has thymine, cytosine, guanine, and adenine, while instead of thymine, RNA is going to have uracil, guanine, cytosine, and adenine. Okay, so I forgot to mention that DNA stands for deoxyribonucleic acid, and then RNA stands for ribonucleic acid. So a nucleic acid is composed of 3 different parts. So one is going to be a 5-carbon sugar, and then a phosphate, and then a nitrogenous base. So in the actual structure, the backbone is going to be composed of the phosphate and the 5-carbon sugar, while the insides contain the nitrogenous bases. So they also differ in location, and that's because the DNA is only inside the nucleus. It's going to stay there, it's not going anywhere, while the RNA can be anywhere in the cell, whether it's in the nucleus, the cytoplasm, anywhere it can be. Okay. Just another difference between RNA and DNA is that RNA is can come in 3 different forms, while DNA is just DNA. So it's going to come in mRNA, rRNA, and tRNA. So mRNA stands for messenger RNA, and that's what's going to make one copy from one protein recipe from the DNA. And then rRNA is ribosomal, and that's what ribosomes are made up of. There is other things too. And then tRNA is what's going to take the amino acids to the ribosomes so that they can build proteins. So I may have mentioned before that there are 2 stages in protein synthesis. The first one is transcription, where you'll be making a copy from the DNA of the mRNA. And then there's translation, which we're literally going to translate that copy into a protein for a shape. So let's get started with transcription. Okay, so I want to talk a little bit about the nitrogenous bases before I really go into it. So as obviously in DNA, there's adenine, thymine, cytosine, and guanine. And guanine and adenine both have 2-ring bases, and they're going to bond with cytosine and thymine. So adenine goes with thymine, and guanine goes with cytosine, and cytosine and thymine are both 1-ring bases. Okay, so I'm just actually going to describe what I've done for my model before I start explaining transcription. So what I have here is I have actually like plasticine backbones. So that's what the sugar and the phosphate are going to be. And then the actual nitrogenous bases are on the inside, and they're represented by those wooden dowels or dowels and the different colored beads. So for adenine, it's going to be blue. For guanine, it's going to be purple. Cytosine is orange, and the other color that I can't really see, I think it's white. Oh yeah, it's white. It's hard to see, but thymine is going to be white. And then on the RNA strand for pink, it's going to be uracil. What are we looking at right now is the DNA and the RNA. The RNA is actually the black strand off to the left corner, but since we're doing transcription, I'm not going to worry about that just yet. So the two red strands that are parallel to each other are the DNA strands. And that when they're actually in their DNA form, they're actually going to be anti-parallel, so twisting around in a helix, double helix. So each side has 4 nitrogenous bases, and like I said, they run opposite to each other. So that's why when RNA is going to go inside, it's not going to copy from the complementary strand, which is the right side, and the template strand is on the left side. So in transcription, you're going to actually firstly be in the nucleus. In here, mRNA is going to make a copy from DNA. You begin with DNA zipped together, and it unzips from helicase in the help of RNA polymerase. The left-hand side of the DNA is a template strand, like I said, and strands on the right side are complementary. Because their nitrogenous bases run on the other side, they cannot be copied, like I just said. So when the DNA is unzipped, the DNA makes an mRNA complement of its entire gene. So when the copy is complete and the mRNA leaves the nucleus through the pores of the nucleus, imagine that I have a nucleus there because it's actually, I didn't draw it, but it has little pores where the mRNA can come in and out. And that's why the mRNA can be anywhere in the cell. Okay, so once it leaves, it's going to go to the ribosomes, and once that's gone, the DNA will zip right back up, and that's the end of transcription. Okay, so now we can move on to the second step of protein synthesis, which is the translation. So like I said, transcription is you're literally making that copy of the DNA, and then translation, you are going to be translating that so that your body or the photosynthesis can create those protein shapes so they can move on to other parts of your body doing what they're supposed to do. So to begin, our RNA needs to make ribosomes. These are thought to stretch out and help reading, and it all happens in the cytoplasm, where the transcription is going to take place in the nucleus. So the ribosomes will advance down our mRNA until they reach the start codon A and U, G, which is adenine, uracil, and G. So this is what this codon is going to this codon is what's meant to start the actual process of making the amino acid chain. So what you see there is where tRNA comes in, and that's what represented is by the crown. So each tRNA will carry on an amino acid, and it's determined by its anticodon. So each one, so this blue crown right here, you can't see what the codon is, but it is AUG for the first one. And so once that's done, it's going to drop off its amino acid, and another tRNA is going to come in with a different anticodon and search for the next set of nitrogenous bases on the DNA sequence, or our mRNA sequence. Like I said, it just keeps going on until it reaches a stop codon, which can be anything from UAA, UAG, or UGA. So the ribosome will fall apart and the mRNA is released, so the amino acid chain folds into its protein shape and is transported. So you see those amino acids right there. So as it goes down, it's going to keep collecting up and create an actual chain of these amino acid proteins. And like I said, when it reaches any of those stop codons, it's going to instantly fall apart. It's going to fold up and make that protein shape, and it's going to leave, and it's going to do its own protein thing. Okay. So that's it for protein synthesis, but it seems pretty organized and perfect, right? Well, it's not always, and that's what's kind of scary about this whole process, because or this like mutation, you could die, and it's kind of terrifying. But basically, what happens, like I'll be talking about just the single base mutations, but sometimes it's the nitrogenous bases that are tampered with, kind of, and change the DNA sequence. So whether it be a swap out of one of the nitrogenous bases, or just a change in the actual sequence itself. Okay, so when this happens, it's called a genetic mutation. And like I said, I'll be talking specifically about the single base mutations on the DNA. And these mutations, they occur for two reasons. So the first reason is that mistakes are made in your DNA, and while DNA is constantly being checked and it's constantly being rechecked, mistakes do happen, and that is kind of terrifying and in that sense. And then the second reason is environmental factors, so things that happen to you that you're exposed to that can cause a lot of trouble. So there's just environmental factors that are known as carcinogens, and so that's going to be cancer-causing agents that disrupt the DNA itself. And they can be anything from UV rays, chemicals, radiation, so that's things like tanning, like causing skin cancer, smoking, lung cancer, just a lot of those, like exposure to X-rays, if you're working in like a specific medical field that requires you to take X-rays, so like vet tech, where you're doing constant screening of dental or just obviously the bodies of animals or people or whatever you're doing. So it does happen, and that's what can alter your DNA code and cause a lot of problems there. So I do want to talk about how these mutations are actually expressed, and that's in two ways. And the first way is silently. Okay, so silent mutations occur in the non-coding parts of the DNA, so they aren't going to significantly alter the phenotype. So the how to explain that? But the genotype that is prominent or dominant, I guess you could say. So it's not going to affect your DNA as much as it would as a missense, or missense mutation. So it generally will just go unnoticed, and exactly what the word is, its name is going to be, it's going to be silent. So then the opposite is going to happen with a missense mutation. So in the missense mutation, this type of mutation is a change in one DNA base pair that results in the substitution of one amino acid for another in the protein made by a gene. So this is actually going to show up in the phenotype, and that can actually cause a lot more problems, and it can be deadly because it's changing the entire genetic code that could. So we could either make something stop working, or things just wouldn't work as normal or as good anymore. Okay, so there's two different types of single base mutations that I'm going to be talking about. The first one is called point mutation, and then the second one is called frame shift mutation. So a point mutation is a single base in the DNA that gets substituted for another base. So what's going to happen is, let's say that you have A, and it gets, all of a sudden there's it gets affected by one of those two factors, and it changes to a G or something. So what's going to happen is it may change what the amino acid is going to come out as, but sometimes it doesn't ultimately change it, because let's say that one amino acid it will code for 3 different types of codons. So if something, let's say that one amino acid codes for UGC, and it has another one that also codes for UAA. So whether that, how do I explain this? One of your bases will get swapped out, but it could still come out as the same type of protein. So that's where it wouldn't become a problem, because it's just coming out as the same thing. So in that instance, it could potentially alter a single amino acid in the chain, but it's not always. Okay, so it is extremely hard to see with this demonstration, because the the nitrogenous bases are really tiny, so those beads but essentially what the second one is a frame shift. So instead of just one single base swapping it for another, what's going to happen is, let's say, so, okay, actually, here's a better explanation. So there's going to be an addition of an nitrogenous base, or there's going to be an addition. Yeah, deletion or addition. So what's going to happen is, let's say that you add one, all those nitrogenous bases are going to have to move over for it. So when you have the mRNA match up with the DNA, it's not going to be the same code anymore. You are significantly altering that entire genetic code in that sequence. So when it comes out to be a protein, you're going to have a whole different set of proteins, and that can be really dangerous and cause a lot of problems there. And then the same thing with deletion. If you remove one, they're all going to have to, literally, it's a frame shift. Everything's going to have to move over, and it's going to code for something completely different. So that's going to change the reading frame of the codons, and every amino acid downstream of the mutation will be affected. And it always occurs. Like when in a gene expression, it's always going to show up, like I said, because it's going to change that protein. So that's it for my demonstration and my just my talk and teaching about what protein synthesis is and how it's related with DNA, RNA, and how genetic mutations occur and happen. And it's possible every single day of your life that you could have a mutation and it's silent and you never know about it. And yeah, it's quite interesting. So I hope you enjoyed this demonstration of protein synthesis, and thank you very much for listening.